Epilepsy is a brain disorder that causes recurrent and unprovoked seizures. It affects over 65 million people in the world, and over 60% of people have no found cause as to why their seizures occur. In order to be diagnosed, you have to have two or more seizures that cannot be explained by another medical reason. The seizure can be of different severities and frequencies. Epilepsy has no definitive cause that has been found, but there are many thoughts as to why it occurs, such as brain injuries, incorrect neuronal development, and illnesses. In order to study an individual's epilepsy, family genetic history is looked at, EEGs are done, and brain scans are used. Seizures are electrical disturbances in the brain that result from a nerve misfiring and sending electrical or chemical signals to the wrong place. These misfires become mixed up and become uncontrollable. Seizures usually cause the body to convulse and for the person to lose full muscular control. However, some seizures do not affect a person as severely and can simply cause someone to stare off into the distance and then regain consciousness. A lot of people with these kinds of seizures go untreated as no one, including themselves, even notice. However, if the seizures are strong enough, they can leave lasting and detrimental effects such as brain damage. Astrogliosis is an abnormal abundance of astrocytes, which are a type of glial cell, that are used to combat destruction or disturbances of the central nervous system. Astrocytes are used to clean up neurons and maintain things such as homeostasis and the blood-brain barrier. The destruction of the central nervous system can be from something like infections, strokes, trauma, neurological diseases, etc. Astrogliosis can cause astrocytes to have changed morphologies and expression. This can ultimately lead to faulty regenerations of axons, changes in physiological responses to the central nervous system, and many other things. Beta-1 integrin, also known as CD29, is a cell surface receptor that binds with other integrin complexes in order to form collagen receptors. When beta-1 integrin is deleted, it can cause reactive astrogliosis. Creatine recombinase is a tyrosine recombinase enzyme that carries out specific recombination events. They catalyze specific combination sites between two DNA sites. This experiment was done to study the effects of astrogliosis on the causes of epilepsy. Some unknowns regarding this field are that epilepsy is treated with antileptic drugs, which work to stop or limit seizures in roughly 2/3 of patients. This means that 1/3 of patients suffering from seizures do not have any treatment to stop them from occurring. As of right now, all the drugs that are available for epilepsy act on synaptic mechanisms that keep neuron excitement at a minimum. In order to find a new medicine that works for everyone, though the goal is to find what is truly causing the seizures, and that maybe instead of the neurons being the only issue, maybe glial cells have a piece as well. Most importantly, astrocytes. For the materials and methods, there are many things that go into this experiment. For starters, the animals that were used were mice because they are thought to be the best model organism for brain-related research in comparison to humans. EEG was used to monitor the brain. Intracranial electrodes were inserted over every hemisphere of the brain, using the left side as reference. Then, hole patch clamp recordings were used to monitor the changes in fluid makeup inside of the brain. Histological procedures were used next. The brain was perfused with PBS and PFA. Then the brains were sliced and had a primary and secondary antibody added to it, raised against IgG. Finally, a Western blot was used in the study. Gray matter was dissected out and was incubated in primary and secondary antibody.
Displayed below are the results of the experiment, some of which included looking at the levels of potassium and glutamate homeostasis in astrogliosis, in addition to neuronal excitability, which characterizes B1 negative mice. Through experimentation, it was determined that mice with chronic astrogliosis do indeed develop spontaneous seizures. The loss of the beta-1 integrins leads to widespread reactive astrogliosis. B1 negative mice had highly abnormal EEG activity compared to the control group. This abnormal EEG activity indicates toxoclonic seizures, displayed in the next slide. It is known that epileptic seizures result from abnormal changes in neuronal excitability. To determine if this was the case in the B1 negative mice, whole cell patch clamp recording was used. It was observed that the neuronal resting membrane potential was significantly more depolarized in the B1 negative negative neurons than the controls. In order to determine if this hyperexcitability was just due to deletion within the neuron itself, tests were performed where B1 integration deletion was neuron specific. In these tests, the deletion did not induce hyperexcitability, and therefore we concluded that it is the astrogliosis that's causing the B1 negative negative pyramidal cells to become hyperexcitable. Using whole cell patch clamp recording, it was shown that glutamate take-up within astrocytes that were B1 negative negative would significantly decrease. In addition, potassium conductance was also significantly decreased. Interestingly enough, protein levels were not shown to have changed. These graphs display the change in time of the protein levels. The data collected suggested that impaired astrocytic uptake of glutamate may cause increased extracellular glutamate concentrations that could actually contribute to neuronal excitability. Conversely, increased potassium currents were found to not significantly alter resting membrane potential. Determined that chronic gliosis is associated with an immature expression profile of neuronal chloride transporters. Western blots were used to detect KCC2 and NKCCL in both groups in order to determine whether CC expression is changed in mice with chronic astrogliosis. Graphs on the next page show that at 6 months and older, KCC2 levels were significantly decreased by astrogliosis. Topics of discussion include a new epilepsy animal model, which discusses elliptiogenesis in the absence of effects from the blood-brain barrier, and indirect and direct effects of astrogliosis. From this experiment shows a strong association between epilepsy and astrogliosis in acute and chronic animal models. Genetically induced chronic astrogliosis was shown to be sufficient to induce spontaneous and recurrent seizures. Previous research indicated that breaches in the blood-brain barrier generated inflammation and astrogliosis. This experimental model studies astrogliosis in the absence of a blood-brain barrier breach, and this further isolates astrogliosis as the main cause of elliptiogenesis. Astrogliosis has some secondary effects, including the downregulation of KCC2 and the upregulation of NKCC1. Reversed ratios of these proteins disrupt balance of excitation and inhibition. Impaired glutamate uptake increased extracellular glutamate concentration and causes hyperexcitability. In addition, reactive astrocytes send off cytokines that signal microglial cells. This signaling back and forth could reinforce astrogliosis after it has already developed.
This study has shown us that astrogliosis may play an important role in understanding what triggers epilepsy, and if we can keep studying the astrogliosis in terms of seizures, maybe a better understanding of the disease will come forward. From this information, we should be able to make new treatments for all types of epileptics and help the quality of life. Overall, the study seemed to cover its bases. However, there were two things that seemed to be missing or could be added for extra results. First, the study did not study edema formation in beta 1 negative negative mice, even though there was a small evidence that edema could be related to the NKCC1 transporter, which plays a big role in astrogliosis. They think that only chlorine accumulation is really to blame in seizures, even though edema mice could show something else and new. Finally, the mice studied were only up to 8 weeks in age. They decided whether or not inflammation or injury was possible causes of epilepsy. This data was overall convincing. However, the age range should be increased to see if through development and adulthood the same data holds true.